In the last podcast, I talked about the methylation cycle, and I went through some of the ideas behind it, some of the inaccuracies around it in terms of treatment, and the fact that there's a lot of information that we don't really understand. And I think it was my very first podcast, I spoke about epigenetics and the aspects of how DNA gets turned on and off. Today, I want to talk about both of those topics together and how they relate, because I think it's one of those pieces that's very important to understand about health and how you can use this in terms of treatment. So stay tuned on today's podcast, Ask Dr. Gil. Ask Dr. Gil. I'm Dr. Gil Winkelman, and as I mentioned in the intro, today I would like to cover epigenetics again. And to cover that, I need to talk a little bit about genetics. So it's one of those things that is near and dear to my heart. I actually did my undergrad in genetics years and years ago at UC Berkeley, as I may have mentioned, and you know, there's this idea that we're hard-coded, and that idea is actually incorrect. So the basic idea behind genetics is that there's all of these base pairs, and that make up our core, and you basically have this long string of DNA, and there's a sequence to it. It's like a code. And that code is basically means the same thing. In other words, you can take a piece of DNA, and it doesn't matter which person that piece of DNA is in. And if the strand of DNA is exactly the same, you're going to get exactly the same thing in each person and in each cell within a person. And the reality is that's not what we're seeing when we look at the research, or what we see clinically either. So to better understand this, I think it's important to talk about what the theory about DNA is currently in mainstream science or mainstream lay science. I was taught 30 years ago that you basically have a string of DNA, and that DNA might vary from human to human, but basically it's the same. And the sequence is hard-coded so that if the same base pairs are in order in every cell, then it's going to produce something exactly the same every time. And there's no defect that will show up with that. And a lot of the theory clinically now around genetic testing is around understanding those deviations that show up. For example, the MTHFR defect that I talked about last time is all about this idea that, there's a change in the base pairing. And as a result of that, there's a change in the expression of the gene. And for some people, and for some conditions, this is absolutely accurate and a fact. For other people, and other conditions, it's completely wrong. And I think epigenetics begins to explain this. So, the epigenetic theory is different than what we've been seeing, and I think it's in part because there is a flexibility in the code. So, as I think I mentioned this in the first podcast, when we first did Human Genome Project, we were expecting to find between 100,000 to 150,000 genes in the human body. And this was based on the level of complexity in people, as well as the level of diversity in people. And they figured, well, there's 25,000 regulatory genes. Well, they didn't even find 24,000 genes in the human body. And since that time, we've actually lowered the number of genes in the body. So now, instead of 23,478, there's roughly 17,980 plus or minus that we see in the human body. And the question is how do we get so much complexity and diversity in people based on that? And if you say that you have a code of DNA and there's a directionality to the code and a start and a stop to the code, you cannot get to that conclusion. So it's troublesome in terms of, what is going on based on the theory. And the thing about it that's kind of maddening is that some diseases, if there is a single nucleotide problem, so let me back up a little bit. DNA is made up of nucleotides, and there's 4 bases. Just to simplify it, there's G, C, A, T, and they'll pair to each other. So G will pair with C, and A will pair with T. And, if there's a change in one of those, then there could be a problem. So the theory behind this is, and this is what the research shows, and this is the piece that you kind of go, huh? Based on the fact that there is a code, but there isn't a code, right? So every 3 base pairs makes up an amino acid, and there's 20 amino acids, and each 3 set of base pairs will then be transcribed into an amino acid, and then you get amino acids joining together, and that makes a protein, and then the protein goes and does something in the cell. And I'm giving you a gross oversimplification of all of this. So, for example, let's say we wanted to make the amino acid leucine. Well, you would use CTT would be coded into leucine. It's a little more complex than that because you actually take the DNA is converted into RNA, and there's the T becomes a U, and we don't need to go into the details of that, but the RNA then makes the protein, and then the protein does its thing. And we've seen this. Like, you look at the RNA, and you get the RNA shows up and forms a protein, and it's sort of been fairly well studied, and you kind of see this. And so the implication is that there's a DNA code, and the sequence makes a difference, and if there's a problem in one of the base pairs, there's a problem in the body, unless kind of like it's not that big of a deal. And I'll give you an example. So the UUU coding makes phenylalanine, okay? The UUC also makes phenylalanine. So if there's a change in the code from UUU to UUC, you don't see a difference. And I'm talking in terms of non-epigenetics here. So the problem, though, is let's say it's UCU instead of UUU. Well, now the phenylalanine becomes serine, and that creates a different protein, potentially, or maybe the protein doesn't fold properly, and so it's not functional. So that's kind of the theory behind why these point mutations called SNPs are creating a problem. And based on what we know about sort of genetics and how genes or DNA is converted into RNA and RNA into proteins, it really makes sense, right? It makes sense that you should be looking at the level of the DNA to figure this out. But I'm going to explain after the break why this isn't what we're seeing. So stay tuned and we'll be right back. Okay, welcome back to Ask Dr. Gil. I'm Dr. Gil Winkelman. So before I continue on this, I just want to remind you that if you have a question or you have a topic you want to cover, please feel free to email me at info at askdrgil.com. That's askdrgil.com. And I am always looking for new topics, to talk about on the program. And if there's something that we talked about that doesn't make sense, let me know, and we'll try something else. So, okay, so let's go back to the DNA and epigenetics. So here's the deal with the DNA and the theory that I just explained, is that theory makes sense if you find 100,000 genes in the body, but really that's not what we're seeing. We're seeing fewer. And so there's more than 20,000 proteins in the body is kind of what we're seeing, but there's not that many genes. And we really don't understand this yet. So I'm letting you know that this does not make sense. And we can't really make sense of it yet. So, but what we do see clinically is that many of the people that we would expect to see problems genetically, in other words, let's say they have an MTHFR defect, and you would expect that problem would show up with a particular type of methylation issue. It doesn't always pan out that way. And, for example, you might see that they should have really, really low histamine as a result of that. But really what we see is that they have really, really high histamine as a result of that. And, not all the pieces kind of fit together yet. But the theory that kind of overlays on this is the epigenetics. And epi means above and then genetics. And this theory was actually proposed in the 1940s before DNA was discovered. In terms of what it looks like. So DNA was known, or at least understood, that something was there, but what it looked like and how it worked, we didn't really know until the 1950s with Watson Crick and their paper that came out in, I think it was 1952. And so the idea is that maybe there's a layer above the genetic layer that's controlling things. And it turns out that theory, 50 years later, was proven to be accurate, at least for the time being. And so the idea behind this is that it's not just the DNA, it's how things get turned on and off. So you could have a gene that doesn't work or a gene that does work, right? And if you have something covering that so it doesn't turn on, then it's not going to turn on, and you're not going to get the piece that you need. And so one of the theories behind undermethylation is that there's not enough of a brake pedal on the serotonin reuptake proteins. And so the body produces way more serotonin reuptake proteins, and those proteins then take up the serotonin really fast, and so you have depressed serotonin, and that's where why, you get a particular type of depression if you're undermethylated that responds well to an SSRI, a selective serotonin reuptake inhibitor. But it doesn't deal with the underlying issue. And if you stop taking the SSRI, then you come back to the same problem. So that isn't that the gene is broken, it's that there's no brake pedal for the gene. And that's a very different sort of approach than just the hard code, well, your code is off, right? Because it means that we can shift that. We just don't know enough yet all the time how to figure that out. And this is where the methylation cycle is so important in terms of balancing the folate and the methylating agents such as L-methionine and SAMe. And my suspicion is over the years, is we're going to be able to pinpoint things based on both epigenetic factors and genetic factors. So, for example, there was a study that came out about breast cancer, and that women who have the BRCA gene, the BRCA-1 gene, typically you're at higher risk of breast cancer if you have that gene. If you take extra B vitamins, you actually can, particularly folate, if you take folate, it can increase the risk of the cancer. So there's something about the folate that is activating that gene more so, is the theory, than just first of all, more than any other vitamin, B vitamin, because apparently if you take P5P or B12, it doesn't affect that. And it can, create more problems and create more incidence of breast cancer if you're taking a high dose of folate. Conversely, other cancers might actually be exacerbated by taking SAMe. And so, in terms of what I do and what Dr. Walsh does, there's nothing that we've seen thus far in terms of the people that we've been treating where their risk factors have gone up. And so I want to just say that generally speaking, this is a very safe approach in terms of treating mental illness. And we don't know why that is, by the way. So the other studies were done in general population with high cancer risk patients to begin with, and their risk became greater as a result of that. There is one caveat that if you are getting chemotherapy, we generally do not do this sort of treatment. We don't want to give SAMe in any dose and folate in a high dose in most cases. So we know that there's some effect there. That's not a population I've seen in my practice. So in any case, going back to the epigenetic piece is really kind of interesting because it totally changes this idea of being hard-coded. And we know that diet, for example, will change epigenetic expression over time. So, not eating processed foods actually helps improve our physical function, but it's doing it at a genetic level as well, on an epigenetic level. And, kind of the way I think about it is that when you're eating really good food for yourself, you're turning on the good genes, right? And we've seen this in terms of meditation and gratitude practices and yoga and exercise. Like, those activities change the way our genes are being expressed. And just because you have a genetic predisposition to breast cancer based on one gene, that doesn't mean everyone with that genetic predisposition ends up with breast cancer. People who take care of themselves and are more careful are at a significantly lower risk than people who don't, do the same. And that's what's so great about epigenetics, is now you have control over your health on some level. Now, obviously, no one has 100% control over what happens to them in life, but at least there are some things you can do to improve your chances and risk factors. And that's the message. I really think that epigenetics really is a hopeful message and empowering message to patients. And I think I'm doing this again because I've had so many patients come in and say, well, I did 23andMe and I am just hosed. Like, there's nothing I can do. And the reality is that's not true. It's not that the information is bad or wrong. It's just incomplete. And I think that having that information can be helpful up to a point. So that's kind of the approach that I take. Anyway, that's all I have for you guys today. If you have any questions, feel free to send me an email. Again, it's info@askdrgil.com, info@askdrgil.com. And I hope you enjoyed this. If you do, please, share it with other people, post it to Twitter or Facebook, and, give a good review in iTunes if that's how you're listening to it. In any case, I will see you next time. Take care and have a great